The overall goal of the hybrid cut tissue sectioning method is to improve the integrity of sectioned recalcitrant plant tissues, those with tough fibers and silica crystals, to create suitable tissue samples for anatomical and biological studies.
Plants that contain high level of crystal and silica can suffer severe paraffin tearing problem during sectioning. This method significantly improves tissue integrity. The main advantage of this technique is that the sectioning under ultra-low temperature in the cryostat hardens the paraffin block, significantly reducing the crystal shearing problem.
The application of this technique can extend to in situ hybridization, fish, and tunnel assays, among others.
Visual demonstration of this method is critical, as the steps to ensure the paraffin block is adhered tightly to the stage can prove challenging.
To begin, select mature Phalaenopsis orchid plants at the 5-leaf stage. Carefully remove the leaves by tearing them along the midrib. Using a sharp scalpel, carefully remove the auxiliary buds from the base of the 3rd or 4th leaf on the monopodial system. Immediately place the samples into a glass scintillation vial containing 15 milliliters of ice-cold PFA fixative. Apply a vacuum to the samples in the fixative at 4 degrees Celsius for 15 to 20 minutes. Small bubbles will be released from the samples. Repeat this step until most of the samples sink after the release of the vacuum. Finally, hold the vacuum overnight and release it slowly the following